All right, so we are going to be looking at mitosis. Specifically today, we're going to dig a little bit into chromosomes and looking at the makeup of those chromosomes today, especially as we look at those chromosome abnormalities that you're doing with your projects. So we know our chromosomes are made of DNA, that deoxyribonucleic acid. All right, and we'll dig into that more later on. But today, we're really going to focus on chromosomes and mitosis, and then later on we'll get into meiosis. Okay, so we know those chromosomes, like we said, are made of DNA. And they're rod-shaped structures, and they are not only made of DNA, but they are also made of proteins called histones. Okay, so you have your chromosome here. You have the P arm, which is going to be the short arm on your chromosome, and then you have the Q arm, which is going to be the long arm. And we say P arm and Q arm because it's going to help us when we're looking at genes to determine where genes are found. So if they're found on chromosome 10, are they found on the short arm? Are they found on the long arm? And then we have a centromere, which is where the chromatids are going to cross. So we know that on our chromosomes, hopefully at this point you know how many chromosomes humans have. So let's think about that for a second. So hopefully you thought to yourself, okay, 46. Okay, so we now have 23 pairs, and we have 22 autosomes, and then 2 sex chromosomes. And on those chromosomes, we found about 30,000 genes that are going to be the directions for building proteins that we're going to get into after spring break. Okay, so we have chromatids. All right, so a big thing that I like to think about when I am thinking about, okay, we've got this DNA and we've got these different versions essentially, is so I go through and I like to think about, we have chromatin. All right, so I wiggle my fingers because the chromatin form is when it is all unraveled. It is just one big pile of DNA. And then we start to twist it and wrap it around our histones because we've got this big pile and we've got to squeeze it all in our cell. And so it's going to form a chromatid. So I like to have people show me your chromatids. So tomorrow, next time I see you, make sure you're able to show me your chromatin and your chromatids. All right. So then before we go to divide our cells, we need to make a copy of our chromatids. So now we have sister chromatids, which form the chromosome. All right. So we have chromatin, wraps around the histones to form the chromatid. We make a copy to form sister chromatids and then form the chromosome. So what's really important to remember about this is because these are copies, when we pull them apart, we don't lose anything. All right, so we need to make sure we remember, okay, these are identical, so we're not losing any genetic information when we pull them apart. Okay, so we talked about our chromatin, and we talked about those histones that wrap them around the DNA, right? And we know we have all these histones because you've got a lot of DNA you got to squish inside your cells. You've got about 6 feet of DNA that is in every single one of your teeny tiny cells. So that's a pretty difficult job. So they use this idea of wrapping, and they wrap it around themselves and then wrap it again and wrap it again. All right, so we have what's called a nucleosome. This is 146 base pairs of DNA that gets wrapped around 8 histone molecules. So this is just the way we wrap it up. All right, then we have DNA, like we said, ends up getting supercoiled in the chromosome. All right, so we end up supercoiling it. That way it's nice and neat and organized, because when we go to divide later on, you don't want just to be trying to divide a big pile of string, or think about your big pile of Christmas lights, it's much easier to separate them if you don't have 3 strands of lights all together. If they were nice and neat and wrapped up, you just had them in piles. Okay, same idea with our chromosomes. We want to make sure that all of that genetic information is going where it's supposed to go. So we wrap it up nice and neat in a chromosome instead of leaving it in its chromatin form. All right, so one of the things that you guys are working on or going to be working on is a karyotype. So a karyotype is going to contain this genetic information. Essentially, it's a picture of somebody's chromosomes. And some of the things that you need to take note of with the karyotype is it can tell us some different things. One of the things that we're looking at is that when we make our karyotype, we are going to put them in order by size. Okay, so we put these chromosomes in order, and they're going to be from 1 being our biggest to 22 being our smallest pair, and the 23rd is going to be our sex chromosomes. So you're going to have that XX or that XY, and it just kind of looks like a little mark there. Another thing that you need to notice about this is we have them in pairs, right? Because remember, where do you get your chromosomes from? So hopefully you know, okay, your parents, right? So one of these came from mom and one of these came from dad, okay? And the big thing about these are called homologous chromosomes. So we know that prefix. Homo means same. So that doesn't mean that they're exactly the same, that they contain the exact same things, but they're going to be the same size, they're going to be the same shape, and they're going to contain genes for the same traits. So for instance, we might have a hair color gene on this one right here, and that hair color gene from your dad may say to have brown hair, and then the one from your mom may say to have blonde hair. Okay, so they can say different things, but they have genes for the same traits. Okay? So that's how we organize them. So smallest or largest to smallest, and then in those homologous pairs. So one of the things that you guys are going to be looking for in your karyotypes, you're going to be looking for if you have a male or a female. And you can also determine genetic disorders based on looking at a karyotype. So sometimes we have extra chromosomes, sometimes we're missing a chromosome, and sometimes we just have an extra part or missing an extra part. So these are going to give us some hints on some genetic disorders we might be seeing. Okay, so here we go, and you can see here on this picture, this is like a computerized picture of what a karyotype might look like. And when you're looking at this, you can see we have dark areas and light areas. So these are going to be our banding patterns. And that's one of the things that you guys are going to really need to look for when you're building your karyotypes. So some of them are going to be similar in size, but you're going to have to look at the details of the banding patterns to help you decide, okay, where does this one match up with? All right, so getting into this next part. So we have mitosis. So we know the purpose of mitosis is essentially to divide the nucleus of a cell. Right, and when we're done, we're going to have 2 daughter cells with an identical chromosome complement. Okay, so we're taking one cell and dividing its nucleus in 2. Okay. So before we can do that, we need to make sure we have a copy. So like we talked about before, we go from chromatid to chromatids that are identical. So when you make that chromosomes and they divide, you're not losing anything. All right, so the first part of this cell cycle, this is technically not part of mitosis yet because we're not actually dividing, is interphase. So this is the stage between cell divisions. I like to think of this as the time in between divisions. Okay, so when I think interphase, I think in between. So this is when the cell does whatever its job is. Okay, so if it's a nerve cell, it's doing its nerve cell job. If it's an immune system cell, it's on the prowl for those bacteria or viruses to attack them. So whatever its job is, that's part of what it's doing right now. It's going to spend a lot of time doing whatever its job is. So we've got some different pictures here, and you'll have an opportunity to see them in real cells as well. So some key things that you're looking for. In interphase, you're going to be able to see the nucleus, and you're going to have the DNA in the chromatin form. So it's just going to be big squiggly lines there. So you've got these pictures right here showing you some interphase cells. All right, prophase. This is the first stage of mitosis. So if you're a pro, you're like the best at something. I also like to think of prophase as preparing, right? When I think prophase, I think prepare. So this phase, we are going to prepare to divide. So one of the things that we have to do, we have to get rid of the nuclear membrane, because right now that's where that DNA is trapped, and we want our DNA to be able to spread out and divide. So in order to do that, we have to get rid of the nuclear membrane. So it's going to disintegrate. Then that chromatin is going to become chromatids to become chromosomes. That way they can get ready to divide more easily. And then the last part here, we have spindle fibers forming. So they are going to come out. They're going to be like spider webs, and they're going to attach to the chromosomes, and their job is to move them. All right, so that's what the spindle fibers are going to do. All right, so here's our prophase cell. And then we've got this. So this is nuclear envelope. It's going to start going away, and we have the chromosomes becoming chromatins becoming chromosomes. And then we have these spindle fibers forming. And then if you're an animal, you may have these centrioles forming. Remember that they're part of the animal organelles that help make those spindle fibers. All right, and then we have metaphase. When I think about metaphase, I think about middle. All right, so those chromosomes, they line up in the midline of the cell or along the equator. And those spindle fibers are going to help move them there, and then they're going to connect to the kinetochore. So that's just part of the chromosome where it's going to connect. All right, so here's a picture of a beautiful metaphase cell here. So we have our chromosomes lining up along the middle. And notice here they are lined up in a single file line. Okay, so those chromosomes are going to line up on the equator in a single file line.